All right. Thank you so much, everybody, for tuning in today and for everybody in the room and from their respective computers. This week's seminar speaker is Dr. Rachel Moore, who's currently a research scientist here at Georgia Tech. Dr. Moore is a microbiologist and astrobiologist whose research has focused on studying microbial processes in different environments, including wildfires, rainfall, arid deserts, and now ice sheets from this past fall, just to name a few of those environments. But today we're going to be hearing about some of her work in the Atacama in Chile. She received a bachelor's degree in molecular and cellular biology from the University of Illinois Urbana-Champaign in 2016, and then a PhD in microbiology and cell science from the University of Florida in 2020. After that, she moved to Georgia Tech, first as a postdoctoral researcher and now as a research scientist. She's a PI of a grant. I think it's called Exomethane, you can correct me, which is a super awesome grant. But anyway, she's now a research scientist here. And with that, Dr. Moore, take it away.
Oh, thank you so much for that introduction. So today I'm really excited to talk to you about this research, life finding in the Mars analog Atacama Desert. And I'm really excited about it because it's not published yet. We're still working on this. So everything I'm going to show you today is still in prep. But it's just a really exciting research, and I'll tell you more about that. So this image here on the first slide is showing South America, and the location of the Atacama Desert, which is within this white box, is in Chile. So if we zoom into that white box, we're looking here now at panel B, note that there's these masses of clouds here on either side of this actual desert portion. So over, for example, the Andes Mountains over Argentina and Bolivia here. And then if we look at panel C, these different colors are sort of shading in different regions of the Atacama. So the blue shaded region is the location of the coastal mountain range. The green shaded region is showing the Andes Mountains and its foothills. And then the yellow is showing the hyper-arid core, or the driest parts of the desert. So the Atacama is known for being the driest place on Earth, the driest and oldest desert. And there are several reasons as to why it is so dry. So I'm going to pop up this little cartoon here, which is kind of like a slice through South America. So there's several reasons why it's so dry. The main reason is because this desert here actually sits between a double rain shadow. So it's sitting between the Andes Mountains and the coastal mountain range that I mentioned earlier. So any clouds that are formed on either side, which we saw sort of initially in panel B, are blocked by the mountains. So rain shadow is exactly what it sounds like. It's just blocking these clouds. Further, there's a Humboldt current is what this is called here in the Pacific Ocean, and it's cold, which makes it less likely for rain clouds to form in the first place. So if they do form here, they are still going to be blocked, or most likely to be blocked by the coastal mountain range. So because of this, because of how dry it is, and also because of the amount of sunlight that it gets, the Atacama Desert is one of the best analogs that we have on Earth for the surface of Mars. So here's essentially a zoomed in picture of the Atacama Desert showing more detail on the precipitation that it gets or doesn't get. So this is a figure from one of our collaborators' papers, Diana Boy from the University in Hanover. And it highlights the hyper-arid, the actual driest part of the desert in orange here. So basically, as you start south in the desert, there is more annual precipitation on average here. And by more, I mean there's around 50 to 150 millimeters for the whole year. So it's still extremely dry. But as you go more and more north into this orange region here, there's practically no precipitation at all. And in many places, it's less than 2 millimeters across the whole year. And this is changing a little bit. In the past, there used to be locations that would go a decade or more without any rainfall at all. That is changing a little bit now. It's climate change. But just for example, when we were here in 2022, we measured in these southern points here around 30 to 40 to 50% relative humidity, depending on the time of day, depending on if we were how south that we were essentially. But we measured in these regions here around 6% relative humidity plus or minus 3% error in the center here, which is historically been thought of as the driest location. So this is just extremely dry. Not only is this desert extremely arid, but it also has the highest surface UV radiation on Earth. And the world's highest ground-based UV index was recorded here to be greater than 20, which is off this chart essentially. So this is a chart showing the UV index for a full year, or the average for a full year from the European Space Agency. So you could see basically in North America here, especially in Georgia, we only ever experience up around to a UV index of 8. I mean, that gets, higher as you go down into Mexico. You can get around to maybe 10, 12, a little bit higher than that. But this down here, this part of South America, you can see on average, it's extremely high. And then, in the Atacama Desert, it could be off the charts. So just to put that into perspective, it's a lot of UV. So this is relevant, of course, as a Mars analog because the surface of Mars receives an extremely high dosage of UV. And it is, definitely higher than the amount of UV that the Atacama receives, but this is the closest that we can get to it essentially on Earth. For the surface of Earth. So microbes definitely need to be resistant to desiccation to actively live here, right? Because it is the driest place on Earth, and probably resistant to radiation, especially if we have any microbes living at the surface, for example. So this image in the background here is actually a picture that I took north of Antofagasta in the Atacama, which is one of our more northern sites. And you can really see in this picture just how harsh of an environment it is and how Mars-like it appears, at least to me, right? There's nothing but rocks and sand, and that's it. There's no plant life, no macroorganisms of any kind. There's nothing larger than a single-celled microbe or spore, for example. Now, a major question here that still persists to this day is whether or not these microbes that have been found in these regions are actually from here. For example, if they are native and specifically adapted to this environment to live here, or if these populations were just transiently deposited from elsewhere, for example, through the atmosphere, and then they would be renewed over time without any significant metabolic activity. So basically things can be blown in with the wind and be deposited, and then they're just basically there dying as they run out of energy because they can't survive in this kind of environment. So this is a really harsh environment, but if even some portion of these microbes are native and actually living here, then understanding more about how these microbes can survive these stressors can not only help us to refine the dry limits of life, for example, but it can also help us to inform future life detection missions in one of the best analog sites that we have for Mars. So that's the hope anyway. For the rest of my talk now, I'll be telling you about my quest to understand more about life in this environment and whether or not we think that life actually lives here. First, I will give you some background on what's been done previously to look at these microorganisms that can barely be detected because they are here in such low abundance. Then I will go into our specific method called our cells first method, which allowed us to generate the first shotgun metagenome data sets from across the Atacama Desert. And then I will actually get into some of our results, which again are still currently in prep. So I'll show you sort of the current status of that. So now we know, since I showed you, that the mean annual precipitation is very low in the Atacama Desert. And the more north we go, the drier it gets, right? This is the same figure, and this is another figure from that same paper. So that is actually shown, the precipitation is shown in the blue dots here. So across the x-axis, we're looking at the different sites. SN in their paper just stands for south-north. That's the south-north transect. And SN1 is their most southern point down here on the map, if you can all see that. And it goes all the way to SN7, which is one of their more northern points, all the way up here on that data set. So these blue points here are just showing the precipitation in millimeters per year. And we can see again that the more north we go, the drier it gets. Well, the interesting thing is that this is actually positively correlated with organic carbon. And that's also shown in this plot, in these box plots to be specific. So we can see that we have less and less organic carbon as we go north until it's almost no longer measurable. It's still there, it's still measurable, but it's just very, very low. And what's also interesting is that in this study, they found that the organic carbon was also positively correlated with extracted DNA when it was measurable. And since nothing else is here, there's no plant matter, nothing else organic, essentially, this has been used as a proxy. This organic carbon has been used as a proxy for microbial cells to give us an idea of the microbial cells that are there, essentially. So I think based on that, we would expect less and less microbial cells as we go north, as it gets drier and drier. That's what we would anticipate, right? Now, nobody has actually measured the number of cells along that full transect of aridity, which again is just going from the very southernmost point all the way up to the northernmost point, because it is, it's a challenge to do that, and there is very few cells there. But this is a really great study from Schultz and McCook from 2018, and they actually were able to measure cells from a couple of different sites, but particularly at Yungay, which is one of our more hyper-arid locations. So this has historically been known as the driest location in the Atacama. That's changing a little bit now. But as we can see here, the thing that I want you to focus on is just that at most, right? At most, we can get maybe 10 to the fifth cells per gram of sediment. So 100,000 cells in total per gram of sediment. And that sounds like a lot, but just to put this into perspective, if we were to go outside here and collect a gram of dirt, you would find billions of cells. So normal soil actually has 10 billion cells per gram. So this is a huge difference, orders of magnitude different, which just makes this very hard to work with because you have so few cells in your sample. Also, in general, DNA extracted from these locations is usually below the detection limit. Even though DNA is there, if you try to extract a few grams from a few grams, essentially, of soil, you will not be able to measure it using a standard fluorometric device. So that also makes it really challenging. And this was a really cool study from Christoph et al. In 2013. They actually collected some sediment from the Atacama. They incubated it with radiolabeled acetate in the lab, which is a carbon source, and they showed that it was slowly broken down over several weeks. So you can see that this is their term for being broken down, essentially. It's mineralization, and they could see that that was increasing. The amount of acetate being broken down was increasing over time. So that shows that some portion is viable, that they're not all just dead, essentially, in that sample, but it doesn't show if they're active in their actual environment, because this was done in a lab, right? Also, although I'm not going to show the data today, previous studies, including Christoph, and then the study on the previous slide as well, have been able to sequence DNA from the Atacama Desert, but they mainly relied on amplicon sequencing, and they only sampled from a few depths and a few sites, which again is of course completely understandable because it wasn't really possible to do true metagenomic sequencing with such low amounts of DNA until now. So I mean, again, just working with unmeasurable amounts of DNA is really tough. Our study, which I will switch over to focus on now, hopefully elevates the current understandings of this extreme environment. So we not only covered the entire hyper-arid core, but we also did collect sediments from multiple depths of a full sediment profile, which I will show you a little bit in more detail. And we also did shotgun metagenomics at every site. So we're now able to look at more than just a couple locations. We're also able to look at all of the genes available, not just the identities of the microbes present, which will give us a more holistic understanding. So the sites that I'm going to talk about today are these 5 sites. And you don't need to really memorize any of these 3-letter codes. They just stand for the site, essentially, kind of like an acronym. So MES is south of Maria Elena, NOAA, NOA is north of Antofagasta, and so on and so forth. But we're going to focus on DGA, BIF, Yungay, NOAA, and MES. These are 5 sites in the north-south transect. Also, I'm going to keep this a little key from all the sites from south to north in the corner here when I'm showing data that are labeled by site in future slides so that nobody has to memorize anything. Again, my main goal today is to determine if life lives here or just persists here. And if they live here, how do they do that? And unfortunately, we're not going to answer these questions today, but I think that we are hopefully starting to etch away at them. And one of the ways that we are attempting to etch away at these questions is by analyzing iDNA versus eDNA. And so what is that? iDNA is intracellular DNA. So the I stands for intracellular, and that is just DNA that's inside of a cell. So this can be a living cell, or it can also be a structurally intact dead cell. It could even be a spore. It just means DNA that's inside of its original body, essentially. eDNA is just environmental DNA. So this is DNA outside of a cell. And the reason why that we're actually interested in this is mainly because DNA in the environment does not stick around forever. It has to be constantly replenished through the turnover of living cells. So just to give you an example of why we're interested in this and how this works, Let's pretend that we're in a desert, right? And we have this single cell here, and for some reason the cell dies, it's lysed, maybe not in this explosive of a fashion, but now its DNA is considered eDNA, right? Because the DNA is no longer fully contained and protected within inside of this cell. So this DNA can go through a few different things. It can just decay abiotically. Another cell can come along and actually take that up, and it can break that DNA down into its parts and use it, or it can even incorporate some of that DNA into its own DNA. Another cell could actually create extra cell, or can create DNases and then export them, and those DNases can also break down the DNA so that it can use that DNA for parts. So in the end, there are many things that are going on to break down and use this DNA. And so in order for there to actually be DNA in the environment, right, eDNA, there needs to be more cells dying and actively releasing some DNA, so that there is DNA constantly getting back into the environment, because it's always going to be broken down essentially. So because of this, it's my thinking anyway that there is a good, if there is a good agreement between iDNA and eDNA in terms of identity, so if there's similar things in both pools, and in terms of relative abundance, so if they're also present in like similar abundances, it should theoretically indicate an environment in which there is a turnover of living cells, like a true environment. So there's things kind of growing and dying, which is what you would expect in an actual environment. All right, so before we can talk results, we need to get to the Atacama Desert, collect samples, and then process them, and I will take you through those steps in the next few slides. So we traveled from the airport here in Atlanta. Here you can see all of our instruments and sampling equipment at the airport. This is Professor Carr here. We traveled from Atlanta to Santiago, Chile, and you could see some vegetation, the mountains here, and some like low-lying clouds. And then we traveled from Santiago, Chile to Antofagasta, Chile in the Atacama Desert. And I just love seeing these two pictures side by side. I just took this out the airplane window. And you could really see the stark difference between, these two locations in the same country. In the desert, we had to use Chilean mining vehicles to get to the sites. So we would travel as far as the road would take us and then travel off-road for a ways to actually get to our final destination. So here I have a little clip of a video. You can see just how bumpy that was. Our collaborator here on the left, Jens Boy, who's also from the University of Hanover, and on the right, that's Professor Carr. You can see him driving us part of the way. And on the left, you can also see some of the outcome of driving on this terrain. So this is showing two of these Chilean mining vehicles. And they both have a flat tire at the same time in these pictures. So you can see some of the amazing undergrads working to replace those tires while we were in the field. Anyway, once we got to the site, we would take a lot of images to document the location. We would take some initial weather readings. This site, again, this is NOAA, which stands for North of Antofagasta. It's one of our most northern sites. It's a really cool looking place. I thought it was the most Mars looking, in my opinion. And I don't know if you can see this on your monitors, but in the background here, this little pinprick of light is actually a solar harvesting tower in the background. So that's one of the ways that the locals can generate energy. So then once we did all of that documentation, we would dig a pit. Also, it's called a sediment profile, essentially. We would dig this down to 60 centimeters in depth. So this is what that looked like at NOAA. This is the sediment profile here at BIF. And we would collect samples from every 5 to 10 centimeters or so, starting at the top layer all the way down to the bottom. So we would do it the same way for every single location. We would take a sample from the top centimeter, and then the 5 to 10 centimeter depth, 10 to 20, 30 to 40, and 50 to 60. And we would take replicate samples from each of these depths from each of these plots. And then we would collect all of those samples into sterile bags, and we brought them back this way to the lab. So once we got to the lab, I spent about 3 months actually extracting the DNA from the cells. And I did this using what we call a cells first method. And I'm going to go in a little bit of detail here because it is pretty different from a regular extraction protocol where you would just take the soil and put it directly into a tube and then extract from that. So we call this the cells first method because we are attempting to take the cells first away from the sediment, and then we can extract from that. So I think that this method allowed us to basically concentrate a large amount of input material. So instead of only working with a gram at a time, I would start with like 40 to 50 grams of sediment, and then I would mix it with some salty buffer and some soapy water, basically. So that's the tween 80. So the reason behind the salty buffer is to keep the cells from popping open, because we want to keep all of our iDNA as actual iDNA. And then the soapy water essentially is just added to work as soap so that we can wash the cells off of the matrix, off of the sediment, essentially. So that's what I'm showing here, is actual Atacama samples being horizontally shaken so that we can hopefully shake as many cells as we can off of the sediment. And then we would spin that down, and then we'd actually take the supernatant, so all of the liquid away from the sediment, and we would wash it another time. And then we would collect that liquid up together in the same bottle. So hopefully what we were left with is all of the environmental DNA and all of the iDNA, the intracellular DNA, so actual cells and spores and such, collected up in this liquid that we washed off of the sediment. Then we would take all of that stuff that was pooled and spin it for a really long time. So that would result in a cell pelletier and then supernatant. So that supernatant would have the eDNA, so all the stuff that didn't pellet down. And then from the cell pellet here, you can see a picture from one of the actual samples. I would extract DNA from that, and then we would concentrate down the eDNA here. And then from both of these extractions here, we just took that and sequenced it. So we sequenced it on a NovaSeq 6000, which is a huge sequencing instrument. And we got about 100 gigabytes of data from this, which sounds like a lot, but across 140 samples, it was only about 500 megabytes per sample. So still quite a lot. It was successful. I was really happy with it, but not as much as you would get, if you were working with regular sample types. Now, because the samples were so low in biomass, I used essentially all of my sample to go toward DNA extraction, and I did not do cell counts myself, which I would love to do in the future to get a real count. However, I was able to estimate the number of cells using the amount of DNA that extracted per gram of sediment. So this is what I'm showing here, is cells per gram of sediment versus the depth below the surface. So I'm showing it for all of the depths. I have all of the replicates, and these sites are just colored here. They're in order from south to north, so DGA down to MES. And what I did was I just estimated that based on the amount of DNA that I extracted per gram of sediment, that each cell would have around 2 femtograms of DNA. So this is probably an overestimate, because some cells can have more DNA than this. But what we're seeing overall is that there are more cells in the southern sites. So DGA and BIF are our southern sites, and there's more cells here versus our northern sites, which are all down here, which is what we expected. We can also see that there tends to be a bit of a trend with less cells at the very top layer and a bit more cells down below, which also is to be expected, because again, there's just so much radiation that they would be experiencing if they were on that top layer. But overall, an extremely low number of cells. We are seeing similar to what's been measured before, right? 10 to the 5th, 10 to the 4th. Our most northern sites have around 1,000 cells per gram, so very, very, very low number. Once the DNA samples were actually sequenced, I got the data back while I was in the field. So this is just an aside for anybody that is doing remote field work or planning remote field work. Bring a Starlink if you can, because I was able to download all of my data overnight and do some really cool outreach. Or maybe, maybe don't bring a Starlink, because it will cause you to do more work. But this is just a picture of the Starlink that I brought outside of my basically the module that I was sleeping in overnight. This is at the Greenland ice sheet, the top of the Greenland ice sheet at Summit Station. So I was able to download all of that data, and then I was able to classify that data, meaning I was able to determine what microbes were there using Kaiju. And so this is just a picture of my setup actually doing that in the big house at Summit Station, which you could see in the background here. That's the big house. And I did this using Google Colab, so I was processing everything on the cloud, basically. So once I classified all of my reads, I was able to see first how the iDNA and eDNA data sets actually differed. So this is just looking at all of the iDNA versus all of the eDNA together, not separating them out by site yet. Surprisingly, the iDNA samples overall were pretty similar to the eDNA samples. So first, 18,000 taxa roughly were identified and found to be present in both the iDNA and eDNA pools. And these taxa represented or were represented in 77% of the reads overall. So the majority of the reads belong to taxa that were found in both iDNA and eDNA pools. This differed slightly, like these percentages differed slightly when I looked at each site and depth individually and compared their specific iDNA and eDNA pools together. But overwhelmingly, the majority of the reads belong to taxa that were found in both iDNA and eDNA for all sites. So according to my logic, this may indicate a group with some amount of activity because cells in the environment would have to be or should be growing and dying in order to have this much shared between these iDNA and eDNA pools. Oh yeah, so these are the individual taxa that were found in only iDNA or only eDNA, and I think that would probably be representing things that just blew in. Now, when we look at all of the metagenomic data and how similar it is by site, we found that 88% of the reads overall belong to taxa that were found across all of the sites. So that is about 7,500 taxa. And to me, I think that this could indicate a core desert group, which in my mind points to there being potential for some indigenous members that are adapted to this environment because they're found across the whole desert. Now, there were taxa that were uniquely found at each site. So you can see these numbers here are taxa that were only found at those specific sites. But those overall, they represented less than 1% of the reads. So they still could be important community members, but they were very low in abundance. Also, we did find significantly higher diversity in the two southern sites. So that's DGA and BIF. But those were also considerably rare and they represented a small percentage of the reads as well. So who is actually here? I'll go into a little bit about this, but not too much into depth just for time's sake. So the most abundant community members were from 8 bacterial phyla, of which the top 2 were actinobacteria and proteobacteria. They accounted for most of the abundance in the data set. So they were about 70, they accounted for about 70 to 80% of the relative abundance across all sites for both eDNA and iDNA pools. And these phyla in particular, it's pretty interesting because they're known to be quite abundant in mesophilic soils. So they're abundant in regular soil, which you might not expect for such a, an extreme environment. However, our communities appear to actually be highly specialized at lower taxonomic levels. For example, we found that a lot of actinobacteria were classified to the genus Rubrobacter, which is known for its radiotolerance, meaning that it can withstand some amount of ionizing radiation. And its radiotolerance may apparently rival Deinococcus radiodurans, which some of you may have heard of. It's kind of the poster child for studying survival in the upper atmosphere, for example. Also notable is that we found the Actinobacterium genus Geodermatophilus, which is known for being tolerant to heavy metals, desiccation, and also ionizing radiation. So a lot of major stressors. This is a representative image of the Geodermatophilus genre from the German culture collection DSMZ. And I'm just showing this because you can see all of these colonies have this really dark, like blue-black pigmentation. And it's actually caused by what is called a melanin-like pigment. So you can see you can see this color and just imagine that this melanin-like pigment does a similar thing for these microbes that melanin does for humans, and it protects them from sunlight. So it's just a useful trait to have probably living in an environment like this. So of the archaea, we again found that the most abundant community members from the archaea were from 8 phyla, and the top 2 were Euryarchaeota and Nitrosphaerota. So the major Euryarchaeota that we found are noted for their extreme halo tolerance, which is pretty relevant for this environment as it is salty, and you tend to find more salt as you go more north where it's drier. For example, we found a lot of Halorubrum and Halococcus, which some of you might be familiar with, sorry. They are genre of Euryarchaeota, and they are halophiles, meaning that they like to live in salty environments. Now, interestingly, the Nitrosphaerota are commonly found in soil also. For example, one of the Nitrosphaerota that we found in high abundance is called Nitrosphaera gargensis. And it is actually particularly known for its tolerance to heavy metals. So this is interesting to me because the Atacama Desert is actually famous for its copper mining. So being able to tolerate heavy metals could be a useful trait to survive here. So some of these Nitrosphaerota can also thrive in low nutrient and harsh environments as well. Finally, we also found eukarya, but they were mostly fungi with some algal reeds as well. So my thinking here is that they were probably blown in spores and algal cells from the ocean, and they're not actively surviving here. They're just dormant or dead or dying. And it should come as no surprise because I've been saying this already, but the overall relative abundances at the phylum level were very similar between eDNA and iDNA, and similar between sites as well, particularly for bacteria and archaea. So I'm showing you here, this is just something, I'm not going to get too much into detail, but this is eDNA for bacteria and iDNA for bacteria, and I'm showing all the different sites here. And then on the x-axis is just relative abundance. So all I want you to really see is the different phyla here, the different colors. You can see that they are very similar in terms of relative abundance between the eDNA and between the iDNA, and also pretty similar as you look between the sites. So just looking at it at the phylum level, you can't really see a huge difference. Since overall the relative abundances at the phylum level look so similar between these sites and between the iDNA and eDNA, I ran some principal coordinates analysis to visualize the distance between individual samples at all of the depths that we sampled at to see if there was any similarities, any clusters. And I found something pretty interesting that was better visualized when I split the southern sites from the northern sites, because there was some occlusion with so many points, but the same pattern is there if you plot it all together. But I'm showing you just here the southern sites first, and then I'll show you the northern sites on the next slide. So this is just BIF and DGA. So we can see when we look here at just BIF and DGA, there's a pretty big cluster here between the iDNA samples, which are shown in the triangles, and the eDNA samples, which are shown in the circles. So the eDNA and the iDNA are clustering for most of these intermediate depths, except for the top layer, which is in this light blue, that's kind of not really clustering, and the bottommost layer, which is shown here in the purple, that's the 50 to 60 depth. So perhaps some of these intermediate depths have some amount of cell turnover, and that's why maybe these samples are clustering based on similarity. So I think that could be indicating some amount of activity. And again, this is just a PCOA looking at the metagenomics data with the abundances for all of the different taxa. Now here is the PCOA with just Yungay, NOAA, and MES. So these are our most northern sites. And again, we can see what appears to be a cluster between the eDNA in the circles and the iDNA in the triangles. But this only happens for the 5 to 10 centimeter depth, the 5 to 10 centimeters below the surface. All of these other samples seem to be grouping by the eDNA or iDNA. So we can see our eDNA cluster here and an iDNA cluster over here in the triangles. So that's pretty interesting. And when I did a PCOA of microbial community function, I actually found the same thing. So I'll tell you what this is. This is a PCOA of normalized gene abundances. So we're no longer looking at a PCOA of just the taxa abundances. This is where I looked at 37 specific marker genes representing different types of metabolism. And now I'm doing a PCOA based on the abundances of these genes for all these different sites. So I've included all of the sites and all of the depths here. So the northern sites are colored in blue, and the southern sites are colored in orange. Now all of the depths are shown in different shapes. So you can see that most of the depths for the northern sites are clustered here, except for, again, this triangle, upwards triangle, is the 5 to 10 depths for MES and NOAA, our most northern sites, are actually clustering with our southern sites. So there's potentially something going on at these specific sort of intermediate depths for our most northern sites in terms of who is there and also what they're doing. And it seems to be maybe something that's different from the other depths. So to investigate this further, I looked at the estimated abundances for specific marker genes that represent trace gas metabolism, which is thought to be important for sustaining other desert communities, for example, in Antarctica, because if you're in an oligotrophic environment, probably the best ways to get your energy and your carbon sources are from gases from the atmosphere. So I found that at the southern sites and at the shallower 5 to 10 depths for our northern NOAA and MES sites that there seemed to be an actual enrichment of two really interesting genes that allow certain microbes to use trace gases. So first, let's look at the gene abundance for the NIF-E hydrogenase. So I will explain to you what this is. This is a cartoon for a heat map. basically just a single square for each site. So you can see I've lined it up with the sites here. With colors that match this key down at the bottom. And I have a single square that's either representing a specific depth or just the max overall. So this key here, what this means, this dark purple means that this gene wasn't either observed in our site or was observed at really low abundance. These lighter or darker, blue colors indicate that this is roughly in the 30 to 40% range for the community having that gene. So for example, that would mean if you were looking at 100 cells, it would be estimated that around 30 or 40 of those cells would have that gene present. And then yellow would mean that every cell in that site has that gene at least once. Okay, so I hope that makes sense. Back to NIF-E hydrogenase. So this is a really cool enzyme that actually catalyzes the oxidation of hydrogen gas. So this organism can use that hydrogen gas from the atmosphere as a source of energy. It's also thought that this enzyme, when coupled with aerobic respiration, can actually result in hydration for the cells. So essentially, as the organism is oxidizing hydrogen gas and aerobically respiring, it can produce metabolic water just as a byproduct of metabolism. And that is retained in the cytosol of the cell based on the direction, the way that this enzyme is facing into the cytosol. So I think that this could be a really important mechanism for cell hydration in the Atacama Desert. Essentially, these cells can make their own water, right, from gas. And interestingly, I found that it is present at a decently high abundance. And by decently high abundance, I mean it's actually in significantly higher abundance than what you would find in regular soils. So it's in about the 40 to 50% range for our most southern sites, BIF and DGA. You can see this kind of bright greener colors right around here. And it was only present at extremely low abundances in the northern sites, except for at this intermediate depth, the 5 to 10 centimeter range. So this is around, I think, the 20 to 30% relative abundance in the community. So it's possible that some microbes in the BIF and DGA sites, and then at certain depths in these more northern sites, that these microbes are actually capable of making their own water from the atmosphere. Again, I think that'd be a very useful trait to have. So another really cool enzyme that we found enriched in gene abundance in these locations is called the carbon monoxide dehydrogenase. So all this enzyme does is it can oxidize carbon monoxide into CO2. So this allows the microbes to use carbon monoxide also as a different source of energy. And then the CO2 can be used as a source of carbon, for example, if they have rubisco genes with the Calvin cycle. So again, I found this present at a decently high abundance in the southern sites, BIF and DGA here, you can see that. And I found it present again at extremely low abundances in the northern sites, except for the 5 to 10 depths. So this looks pretty low here, but it's about a 20% estimated relative abundance. So again, that it is pretty low, but it would perhaps be another way that the microbes here in these northern sites can be making energy and making carbon more available as well. So another really useful gene. Finally, something that I did not find much of at all were marker genes for phototrophy. Even though we did identify cyanobacteria and algae in the metagenomes, they were at low abundances, and so were their specific phototrophic marker genes. For that reason, I do not think that phototrophy is the main form of primary production in this environment. And I think that's pretty relevant given the talk that we had last week from Dr. Dutta, who was talking about primary production in the dark biosphere, right? This is kind of a similar dark environment, so I think that makes a lot of sense when we're talking about all of the deeper layers. I think that trace gases in this highly oligotrophic environment are more likely to be important here for producing energy and also maybe for making carbon more available, for example. So in summary, this is my final slide. The Atacama Desert may harbor more diversity and potential indications of activity than previously realized. And I must stress potential indications because we did not actually measure activity. We're just looking at some signs of activity, essentially. I also think that trace gases they probably serve as an important source of energy for Atacama bacteria, also probably for carbon, because a lot of our, I didn't show this on any slide, but a lot of our, 100% of our, sorry, I'm saying this wrong, but we found that at MES, which is one of our most northern sites, actually had a very high abundance of the rubisco gene, which is used to actually fix CO2. So about 100% of the cells there are estimated to actually have the rubisco gene. So I think that CO2 could also be really important for carbon. So basically, these microbes, I think, could be primary producers, the ones that are actually taking these atmospheric gases and using them to make energy, using them to fix carbon. And then certain depths, like I've showed you, are probably important. Probably important places to look for activity, and I think that they are of particular interest for future work. And then I would like to just thank everybody that was a part of this project that made it possible. So Christopher Carr is the PI on this project. He's my advisor as well. I work in his lab. Jens Boy, Diana Boy, and Armando. So thank you to all of them, and I hope that you enjoyed my talk. Thank you for listening.